learning objective include oxygen requirement or oxygen is not needed or needed, and microbial classification based on oxygen requirement, and also how various bacteria have evolved to deal with harmful substances that are made by oxygen. Well, oxygen is needed for aerobic respiration, and those bacteria that use oxygen in aerobic respiration, they can synthesize maximum ATPs. In other words, if a bacterium is able to use oxygen, it means that it has more ATP available with it. As you can see in the previous lectures, we mentioned that when glucose is metabolized through glycolysis, it produces a pyruvic acid, and that could be channeled through Krebs cycle, creating NADH and FADH2 molecules. These electron carriers then can be put into electron transport chain, and this process at the end by chemiosmosis produces about 36 or 38 ATP molecules. It's a lot of energy. So those bacteria that utilize aerobic respiration using oxygen, they have maximum ATP synthesis ability. On the basis of oxygen requirement, we can classify organisms into various groups. We have obligate aerobes. These are the ones that need oxygen for their growth. If oxygen is not available, they won't grow. So these are called obligate aerobes. Example is a Mycobacterium tuberculosis, the organism that causes TB in humans and animals, is the one that requires oxygen for its growth. The other group is called facultative anaerobe. These are the organisms that they basically like oxygen, presence of oxygen, but if the oxygen is not available for some reason, they don't mind it. They keep growing. So these are called facultative anaerobe. We can differentiate these cells based on a test that if we use a liquid medium and in a test tube, if we look at their growth pattern, you can very easily see that an organism that likes the presence of oxygen would grow maximum, or it would show maximum growth at the top of this medium, because oxygen is more prevalent here than it is here. So these dots basically show bacterial growth diagrammatically. You can very well appreciate this is an obligate aerobe. On the other hand, here it's a facultative anaerobe. As you can see, the growth pattern here at the surface is more, because obviously oxygen is more here. So this guy likes oxygen presence, so grows more, but you can see clearly that this is obligate aerobe. It only is present at the surface, but the growth is almost throughout the medium. So whether oxygen is available or not does not matter to it. But because here it prefers growing in the presence of oxygen, it has more growth here than anywhere else. But still, there is a growth evenly distributed. And E. coli is one of the examples that is a facultative anaerobe. Now, oxygen basically is harmful. It produces oxygen molecular oxygen can combine with an electron and convert itself into a superoxide radical. And these radicals are really crazy molecules. They can combine with any enzyme or any protein and can damage it. So the product of oxygen basically are harmful for the bacteria, and bacteria have evolved themselves to get away from these radicals. As you can see, that oxygen can produce hydrogen peroxide, and hydrogen peroxide can further be degraded, converted into hydroxyl radical. So all these molecules, they are harmful for the bacteria. But some bacteria, not all, but most of them, they have evolved themselves to deal with these various superoxide radicals and all that, hydrogen peroxide. As you can see, there's an enzyme called superoxide dismutase. Many bacteria have it. So they can convert these toxic substances of oxygen into just oxygen and hydrogen peroxide, And hydrogen peroxide can further be degraded by the enzyme catalase into water and oxygen. So this is the way they deal with these toxic oxygen metabolites. And all of them have not this ability, we'll see in a short while. The ability of some bacteria to utilize or to convert oxygen metabolites into harmless molecules could be tested by a test called catalase test. What we do is we take a drop of hydrogen peroxide and put that drop onto the slide and take a colony or few bacteria that we want to test and mix them together. If the bacterium has this enzyme catalase in it, it would use this hydrogen peroxide and convert this into water and oxygen. And oxygen would form these bubbles. As you can see, small bubbles there. And that indicates the presence of catalase in this particular bacteria that we tested here. As we were talking about the oxygen requirement and the classification of bacteria based on oxygen, another category, the third category, is obligate anaerobes. These are the ones that do not like oxygen, or they do not use oxygen at all, and the presence of oxygen is toxic to them. In other words, they do not have a mechanism to dispose of those toxic substances that are formed as a result of oxygen metabolites. And if you see in the tube, in the liquid medium, their growth pattern, they will be sitting right at the bottom because here there is no oxygen. Another group is aerotolerant aerobes. These are very interesting microorganisms. They do not use oxygen at all, but they do not mind the presence of oxygen as well. So their growth would be evenly distributed. Whether oxygen is there or not, they do not use it, but they can grow happily even in the presence of oxygen. So they do have a mechanism in place to dispose of oxygen metabolites. So they are called aerotolerant aerobes. And very good example is a bacterium called Helicobacter pylori. This is a bacterium that is present in the stomach of animals and humans also, causes gastritis and gastric problems. Another category of is what we call microaerophiles. They're also very interesting organisms. As you can see, their growth is confined to an area, a small area in the tube. So they basically like oxygen, but they do not like too much of it. They like a little bit of oxygen. And that is the reason, because oxygen is more prevalent here than it is in the middle of the tube. So they like low tension, what we call, they use oxygen, but the oxygen requirement is in low amounts. They produce, in other words, we would say that these bacteria, when oxygen toxic substances are generated, they are more generated if they grow here. So those organisms that grow here, they cannot handle those that many toxic substances from more oxygen. So that is the reason they go a little down where oxygen is not that much prevalent. And also, it is important to know that they're not anaerobes. So they do like oxygen, but in low concentration. Listeria and Vibrio are examples of those kind of bacteria that like low oxygen tension. In summary, those bacteria that use oxygen, they make a lot more energy, ATPs, than the others. Anaerobes do not use oxygen. They don't like it because they cannot grow. The toxic substances that are present in oxygen can kill those bacteria. And a very good example is Clostridium tetani that causes tetanus in humans and animals. We saw that organisms could be classified on the basis of whether they use oxygen or they do not use oxygen into various groups.